First exposure to ME/CFS came when my 14-year-old daughter had an attack of glandular fever, and then soon after that had a rapid down spiral of health, which involved becoming allergic to every food item she took in her normal diet, and also pervasive fatigue. So as we do, we went to our health practitioners, and I have to say this was in 1990. They had no idea what was going on. And so, as I found, senior clinicians, when they go through all of their toolbox and they can't find anything obvious, they just invoke the default position. So they told us this has to be psychological or psychiatric. So I was, I have to say, a bit surprised, because a year or two earlier, there'd been an outbreak of an unknown disease in a small Otago town near Dunedin called Tapanui. You may have heard of this. And the GP there, Dr. Peter Snow, made quite a fuss about this. And in fact, it got written up in the New Zealand Medical Journal. And of course, it's now in our folklore, health folklore, as Tapanui flu. But I have to say, what struck me about this illness was the pervasive fatigue. My daughter had been a highly active young teenager and suddenly went to being able to walk for 5 or 10 metres and then having to rest for 2 or 3 hours. So as a biochemist, that said to me, there had to be something wrong with the energy production in her body. And it so happened that a young academic joined my department just at that time, who was an expert in energy production in humans. His name was Dr. Mike Murphy, and he developed what you may know of as MitoQ, which is available now in pharmacies. And so we put in a grant to the Health Research Council to start. I wanted to do research on this and find out what was going on with my daughter's illness. And the response we got from the clinical reviewers is that time, this is not a real disease and you won't find anything interesting. So my desire to do research on it actually really went fallow for 20 years, and it was, after that time that I managed to start working on it. So in the meantime, once we got a diagnosis of ME, I looked into the history of this and found that since the 1930s, there's been about 75 reports of outbreaks of infectious disease that has ongoing illness. And the first in my lifetime, so I've just listed 3 in my lifetime. In Northern Iceland in 1946, it lasted about 3 years, there was a report of about just over 1,000 cases, and I won't try and pronounce the name of the disease, but that's the North Iceland town. And interestingly enough, in 1986, 40 years after, researchers went back to try and trace some of these people and asked them if they still had disease, and 80% of those they found still had ongoing disease. So it seems sort of classic ME/CF in hindsight. Perhaps the one that had the most publicity was from the Royal Free Hospital in London in 1955, and interesting that Dr. Ros Vallings actually was a young doctor in training in London at the time, and actually helped in looking after the people at that hospital who came down with this mystery illness. And it tells me it was like a flu-like illness, I think you said, Ros. So that's where, and 292 cases. I just want you to note the number of cases there for these. 292 cases, and that's where the name ME came from, myalgic with pain and encephalo involving the brain and then the inflammation. And then near to the time when we had our Tapanui flu, there was a small village on the shores of Lake Tahoe in Nevada, Incline Village, which reported a strange outbreak. And the 2 GPs there were inundated with people coming with pervasive fatigue. So they got the Centre for Disease Control over to do an investigation, and they went back and wrote a report, as they do, and they reported that it was mass hysteria. So that's kind of where we've got to. Now, I should say that, as Ros, I think, pointed out, it's not only viruses which give rise to ME/CFS. And I wanted just to tell you a little story about an 86-year-old woman from a Hamilton rest home that wrote to me recently. And she had a colostomy at age 43. 43 years ago, and she immediately went into an ME CFS state, and quite a severe, she was homebound, etc. And she was so excited to send me this email because she read in the Ostomy New Zealand magazine, I'd never heard of that, but it's for people that have had colostomies and other similar surgeries, that people who've had this should take high levels of zinc each day, and she'd never done that. Quite a lot of ME people, of course, do take zinc supplements. And she took this, and she said it was amazing. My symptoms almost completely resolved after 43 years. So I responded to her and said, can I tell the third-year medical students about this? And she was very excited that she was going to go down history with the medical students. So that's a good news story about someone finding something which helped their illness. So what was the dominant voice during the time when my daughter got her illness, and the time really through to nearly the time when I actually started thinking about doing research on the disease? And it was this psychiatrist in the United Kingdom called Sir Simon Wessely, who was president of the Royal Society of Psychiatry. He was a very dominant figure in health in the United Kingdom, and he spent 20 years working on ME and feels it's the best work that he's done in his life. But he concluded that it was a perception disorder. That is to say, ME people perceive their symptoms and perceive they had a disability, and because of those perceptions, when it came to anything in life, they perceived that it was too hard, so it was an opting out illness. And yet he acknowledged that the illness started with a viral infection. So for me, I couldn't understand how you could make that initial conclusion and then come to the fact that it was a perception. Not surprisingly, families in the United Kingdom were very distraught and angry with this kind of diagnosis. And in fact, he started getting death threats after the turn of the millennium, and stepped away from working from the disease. But that was, a dominant thing. So what about in New Zealand? And I'm going to tell you my 30-year history as a parent of an ME family more than as a researcher. And so, of course, I said we were told that my daughter's illness was likely to be psychological or psychiatric. And I have to say that it's really good that there's slowly been an improvement in attitudes about this illness over the 30-year period since I've had an exposure. A lot of that is because Ros has done a lot of education with GPs, and so people now have a lot better perspective about this disease. However, just recently I've had a couple of queries and requests from people with ME, would I write them a supporting statement because they've gone down with the illness, they've had income replacement insurance, and the insurance company has hired a psychiatrist from Auckland to consult on the case, and he said, no, this is just a perception disorder, and so they've been denied payment. Isn't that just scandalous? And so it's still happening today, and I mean that, I don't get angry often, but that made me so angry when I got the first one. I took Easter Monday and wrote the woman a 6-page report, and she wrote back and said, but you don't even know me. Okay. With our research recently, because we've managed to find some molecular signatures for ME/CFS, and I talk about as finding the biological basis of the disease, but the media don't like that, so they replace my headings with ME is a real disease, it's not psychosomatic. I would never kind of use that language, but it's kind of a good language to communicate, I think, to the community. So I've then had a flood of emails, the longest emails I've ever had from ME patients telling me their heartfelt history and saying no one's ever affirmed their disease before. So, that shows that the pent-up sort of stuff out there in the community with people that have suffered from disease, some of them for as long as 40 years, some of them only for a year or two. There'd been no medical school training of this disease, so medical students going through had never been exposed to even the concept of a disease like ME, and so going out to practice, of course, when they meet it for the first time, you can understand how difficult it is for health practitioners. So in 2013, I was invited to give a guest lecture to the third year medical class. And I have to say, the students responded to this lecture far better than any other lecture I'd given to medical students in the past. And so this was in an unexplained disease module, and I've been invited back every year since. So now I've actually lectured to about 2,000 medical students. It's probably the best thing that I've done for ME, I hope. So there'll be groups of graduating students coming through, which at least will have an idea about this illness. Now, I got up there, social services not accepting patients cannot work or need help. What I mean by government social services, I don't mean a social service that's afforded by this organisation. And unfortunately, I had a little bit of discussion about this yesterday when I met Amanda and Charlotte, and we were able to talk about that. And what I mean by that is that when a few years ago, of course, WINS wanted people on sickness benefits to actually have a designated doctor give them a consultation to assure that they needed to be on that. And I know several ME patients that went had a 7-minute consultation. Some of these had been housebound for over 20 years, and the designated doctor said there was no reason why this person can't work full time. And so they lost their benefit. And again, that's another sort of tragic thing that's been going on. And families, of course, we all know it's so difficult for families to understand this disease. And when I was last in Tauranga, it was 2013, again with Ros, we did a workshop together. A young girl, the first time I talked about my daughter's illness and our family's interaction with the medical system and how difficult it was. And a young girl came up to me in tears and said, you know, your daughter is so lucky to have a father like you. I kind of cringed and was embarrassed. And I said, why do you say that? And she said, because my father just doesn't accept my illness and just tells me to get on with life and stop complaining. So, you know, you understand how difficult this disease is for families. So none of this is meant to be a criticism of anyone. It's just saying this is such a complex, difficult disease. It's difficult for patients. It's difficult for families. It's difficult for health practitioners and, not surprisingly, government social services. And you know, that's what we've lived with. Now, the landscape for ME/CFS has changed, of course, dramatically with the COVID outbreak. And, you know, I'm just kind of feed into Sarah's talk. We now have over 100 million people infected with SARS. So here's a virus which was always likely to give rise to ongoing illness. But the outbreaks I talked about in my early slide was hundreds, or the most, a few thousand patients. Even the initial SARS epidemic in 2003, there were only 10,000 cases worldwide, or less than 10,000. We have 180 million, and that's why this is such a serious illness, because, as Sarah mentioned, it's likely to throw up worldwide a huge number of new cases of an ME/CFS-like illness, maybe ME/CFS. The difference is, of course, this illness, the long COVID, is caused by just that one infectious disease, as distinct the ME/CFS we know is from a number of infectious diseases like glandular fever and whatever, and also other causes. So it's likely to have somewhat more be a more specific subgroup. So what's the reaction to that been? Well, what we've seen is there's been a high degree of attention from the clinicians who've been dealing with the SARS pandemic from this. They've been really interested in these responses. And many of them have been surprised. You might say they shouldn't have been because we've got this history, but they were. And the interesting thing is now we have social media. So the so-called long hauler group in all countries have formed their own social media group, including New Zealand. So Sarah mentioned, you know, statistically you would expect maybe 137, was it, the number you used? In the New Zealand long hauler social media group, there's 200 to 300 people now. So that's running, if you like, about 10% of the infections that we've had. And of course, they're therefore a powerful group that can advocate for themselves, and that's a kind of a new phenomenon. ME never had this for a long period of time, and I'll come back to this in a minute with ME. And as Sarah, I think, mentioned, special units are being set up in the UK to study the various subgroups of what's thought to be there in the long COVID group. She mentioned also that NIH has promised, I've got there over $1 billion, but we should say 1.15 because that 0.15 is another $150 million. And when I get in 2 slides to talk about the funding for ME, you'll see how hugely different that is. And in New Zealand, the Ministry of Health, again with the long hauler social media group, has been advocating. They have got a fairly modest funding round going at the moment to support this area. So what I'd like to do now is to show you a short video of one of the most influential women with ME in the world, Jennifer Brea, who co-founded a group called ME Action, which has lobbied hard politicians and funding in the United States, and also made a long documentary of her own illness. She filmed her own illness, and you may have seen that. That's the film Unrest, which came to New Zealand in 2018 in our International Film Festival. I've got a couple of minutes here that comes from a TED talk that she gave, and this is I show this to medical students because it's a very powerful 2 minutes which tells them what the disease is really like, and she's a very good advocate. So,
hi. Thank you. So, 5 years ago, this was me. I was a PhD student at Harvard, and I loved to travel. I had just gotten engaged to marry the love of my life. I was 28, and like so many of us when we are in good health, I felt like I was invincible. Then one day I had a fever of 104.7 degrees. I probably should have gone to the doctor, but I'd never really been sick in my life. And I knew that usually if you have a virus, you stay home and you make some chicken soup, and in a few days everything will be fine. But this time it wasn't fine. After the fever broke, for 3 weeks I was so dizzy, I couldn't leave my house. I would walk straight into door frames. I had to hug the walls just to make it to the bathroom. That spring I got infection after infection. And every time I went to the doctor, he said there was absolutely nothing wrong. He had his laboratory tests, which always came back normal. All I had were my symptoms, which I could describe, but no one else can see. I know it sounds silly, but you have to find a way to explain things like this to yourself. And so I thought maybe I was just aging. Maybe this is what it's like to be on the other side of 25. Then the neurological symptoms started. Sometimes I would find that I couldn't draw the right side of a circle. Other times I wouldn't be able to speak or move at all. I saw every kind of specialist: infectious disease doctors, rheumatologists, endocrinologists, cardiologists. I even saw a psychiatrist. My psychiatrist said, "It's clear you're really sick, but not with anything psychiatric. I hope they can find out what's wrong with you."
We can put that onto.
yeah, it's a very compelling 2 minutes. She was a PhD student of Harvard when she got her ME/CFS, and you know, she just really illustrates what the disease is all about. So she was responsible, really, and her organisation, for changes in attitudes to ME/CFS in the United States. And so in 2019, just before the pandemic, the Senate passed a bill declaring that ME/CFS was a serious illness, and it declared that actually health practitioners and social services had not done well with patients with this illness and their caregivers in the United States, and it attested that they would do better in the future. And it even said research support had not occurred, and they promised that the National Institute of Health would actually give money to this. And I was so excited. I saw a few $1 million. And so I want to contrast that, what they're saying with long COVID now, this National Institute of Health offering $1,150 million for long COVID. And that's got to be good in the long term for ME from what's found out of that. The very good thing that happened in 2019 was that 2 research centres were set up, one at Harvard and one in California at Stanford. And what that meant is mainstream researchers were coming to work on this illness. And so that was a huge sort of shift, if you like. So even before COVID, I was enthusiastic. We were going into a new era of understanding, but with the long COVID and the increased attention and funding, you know, I'm even more so. Okay, so when I was contemplating doing, getting going on research in this, I'm a biomedical researcher, and a biomedical researcher tends to look at a problem in a very focused way and in a very detailed way. I work on Alzheimer's disease as well, and I'm working on a human brain protein which is neuroprotective as a potential therapeutic for Alzheimer's disease. That's a very focused project. I looked at all of the research that was in the literature, and it was kind of exciting vignettes of research, very focused. But it was like pieces of a jigsaw puzzle. So when you look at that puzzle, I don't think anyone in this room could work out what the solution to the puzzle is. I'll show it to you right at the end of my talk. And it struck me, this wasn't, my normal way of research was not the right way to approach the disease. We had to do it in a much more holistic fashion and kind of look at the overall aspects of the disease. So influenced also because I was a parent of an ME/CS family. So the research questions that I wanted answered was, what is the major control centre in the body that actually causes all of these severe and diverse and long-lasting symptoms I'd seen in my daughter? You know, why doesn't the disease get better? Why didn't my daughter get better? So I wanted that question answered. And although 3 in 4 patients move from an acute phase, my daughter had acute phase where she was bedbound and housebound for about 4 years, and then moved in where she, you know, could go into a vertical position and have some sort of interaction out with the world. When they go into this, they're also very subject to very frequent relapses. And so I wanted to know what actually drives this relapse recycle, and if this time I'll show you our research on that at the end. And then there was another question added on the 25th anniversary of my daughter's illness when she was aged 39. She rang me up one morning in my office in Dunedin and said, "Guess what, Dad?" And I didn't dare say what I thought because I knew she desperately wanted a child, and she said she was pregnant. And the amazing thing about that wasn't just that I was going to get a grandchild, it was actually during her year of pregnancy, she had the best year of her ME life ever. All of her symptoms just regressed. They were still there, but her quality of life improved. She really loved her pregnancy. And so, you know, we can ask her, well, why I was so excited about this was because it said to me, the symptoms of this disease and the disease itself is not irreversible. It should be reversible if we can just find how to reset the homeostatic point, which has been sort of shifted with the disease. So you could say, and I think Roz mentioned this, you know, what is it about pregnancy that might cause this? And it can be, there's an increased blood volume, suppression of the immune system, and of course hormone stability. But in our case, what we know really caused it was this person here. So this is him at 8 months old. He was a healthy little boy there, and you can see I provided him with some clothing just to make sure he knew what his future was. I have to say now, he's just 6 now, and I call him a preteen. And he's mostly healthy, but he is very prone to viral illnesses. So I'm going up to look after him for a week in a couple of weeks' time, and I hope he'll be free of all his illness by the time I come up. So, and that's been a real joy, not only to my daughter, but also to me. Okay, so how then, I said I didn't want to just do what I normally do, very focused biomedical research. I wanted to do research which on these big questions, and how would I do that? And I was inspired actually by a geneticist at Stanford University, he's now head of genetics there, called Michael Snyder. And he did a very self-indulgent study, cost a couple of $1 million. He had blood taken from him over a period of 2 years, and he did every molecular test you could do, what we call omics, very complex molecular studies on what was going on in his body over the 2 years. He had a common cold during that time, and he had a fairly mild upper respiratory infection. And, you know, not very much, but it was amazing to me that you could see all of the molecular changes that were going on during the event and then resolving. And I thought, this is just what we need to do for ME/CFS. So this is what I decided to do. However, you can see, even just working on himself, he spent several $1 million, and the paper had 40 authors on a Cell paper, which is a very high impact paper. So how would we do that then with a limited budget? So I went to the rare disease literature, and I've got an ex-student who has helped to form a company called Memento Pharmaceuticals in Cambridge, Massachusetts, and they work on rare disease. And he was very encouraging. He said, this approach that we use for rare diseases would really be perfect for an unexplained disease like ME/CFS. And we used to call this personalised medicine. It's now called a precision medicine approach. And I have to say, to do this, you need an expert clinician. And I'm just forever grateful for someone who you may have heard of before, for what she's contributed to my research. She diagnoses all the patients in my study groups. She just gives valuable information. And as she said, she's written a few books. And, you know, I can't thank her enough. An amazing woman. Okay, so we had to be strategically very careful to make the dollars go round. So all we did, we took 20 mils of blood from each patient. We separated into the plasma, the immune cell fraction, and neutrophils, which go down with the red blood cells. That's the red bit at the bottom. And we've just done multiple studies on that kind of one sample from really each patient. And my technician, who started with me at age 18, she's just had her 60th birthday. She's fractionated so many blood samples since we started. And we've piloted a diagnostic test for a molecular biomarker. We need more money and bigger patient groups to actually do that. We've done work with plasma, but I just want to give you now some vignettes of the research that we've done and the results that we've got looking at these multi-molecular big picture studies that Michael Snyder did on himself. And they're called, that's of the expressed genes, or transcriptome as it says there on the slide, of the proteins of the immune cell, of the energy production, and more recently, what's going on in the DNA. So I'll try and just get through those. If you're all slumping over your desks before I do, I might stop in the middle of it, but hopefully I'll get a chance to tell the complete story. I could talk for hours about this, but I'm trying to be very disciplined. So the first thing we did was to look at what is happening in the activity of the DNA in expressing information. This is called looking at the active genes or transcription, production of RNA, in fact the messenger RNA, which is like the messenger RNA in the Pfizer vaccine. And so my PhD student Erin Sweetman looked at 13,000 transcript from each patient. And I just want to show you the top 3 that were different and ME people compared with age, gender matched controls. And we used 10 patients and 10 controls. So that's our limited group, so we could afford this. And we found the top 3 were involved in inflammation. The top one change was an inflammatory cytokine. That's an immune regulator molecule, IL-8. But interestingly enough, the other 2 were anti-inflammatory. So what the body was trying to do in ME patients, we saw just from these 3, was that there was inflammation, as Roz has mentioned, probably destructive inflammation, but the body was trying to counter this by raising things which would be anti-inflammatory. So that was a very, a good sort of insight into understanding the disease right from the beginning. And then she looked at all of the wealth of data that came out of this, and we can do now what's called pathway analysis. We can look at the changes in individual transcripts and relate it to what does this mean to the overall physiology. So these in red are the things that we found were disturbed in ME patients. Energy production, what we call mitochondrial function, because I'll show you this in a minute. Mitochondria are the bodies in the cell which actually produce the energy. Oxidative stress. Now, oxidative stress comes because when we're actually making energy, we produce reactive oxygen species called ROS. That's not the same ROS that you've heard of. And the body has to control that, otherwise, that's damaging to our health. So we saw there was dysfunction in that control. We saw a lowered rate of metabolism, and that's now a general understanding worldwide that people with ME sort of go into a almost like a hibernation state, a lowered level of metabolism, which we think now is a protective state. The body senses it's in danger and goes into a protective state. And as I've said, we saw inflammatory pathways, immune pathways, those were expected. The one we didn't really expect, perhaps we should have, was that the circadian clock, which determines our circadian rhythm, was all disturbed in ME. And why we should have realised, because the circadian rhythm is important for sleep. That's a major symptom. But actually, turned out when I read into the literature, people with a disturbed circadian rhythm have all of the symptoms like ME. And so, in fact, that alone could actually explain all the symptoms. I'm not saying I think it does. I think it's a combination of things. So that was the results of the first study. And then Erin stayed on with me after her PhD had finished to do a postdoctoral period, and she looked then at the immune cell proteins called the immune cell proteome. And so what she does is she isolates the proteins from each patient control and digests them up into small fragments, and then she pulls a little bit from each of them and build what we call a spectral library. That means that so we can determine all of the proteins that are in the patient and control groups that we can match against. And then she did a very sophisticated mass spectrometry approach to classify the proteins in the patient and control samples. And we get huge amounts of data, and so we try and stratify that by doing what's called a principal component analysis. So you're getting really sophisticated research data here. So I hope you'll tell your grandchildren about this. And what we were doing here is I was wanting to see whether in fact the ME group would separate from the controls. And the ME patients are the red dots, and the controls are the blue dots. And even visually, we could see that 7 of the, in fact, it was 11. I don't know how we had 11. It was supposed to be a 10 and 10 study, but we ended up with 11 ME patients in the study. 7 of the 11 just visually separated. And then I got a biostatistician in to actually look at the data carefully to make sure a visual thing like this was appropriate. And he came back and said, actually, when you do a linear regression analysis, you can separate 9 of the 11. And so that was quite exciting for us, that the ME group separated on the immune cell proteins, because that said there's a molecular signature in there. And so we were interested to identify that, and it turns out we found a lot of the proteins, as I'll show you, involved in energy production were dysregulated in the amounts in which they were synthesized. They were either synthesized in greater amounts than normal or less. So normally when you're synthesizing proteins that are needed for cell functions, they're controlled in very delicate ways so you get just right, the right ratios of everything. And we're saying this is dysregulated. So I want to just give you a little bit of first year biology thing here. This is a human cell. It could be an immune cell. And if you can see those biscuit things in the, those little purple biscuit things that look like they've got squiggly icing on, those are what we call mitochondria. Those are the factories where energy is produced. And just on the side, you'll see a little yellow arrow, a little squiggle with ATP next to it. That's actually the energy molecule of the cell. It's like the currency of the cell. And if you look at those, the right-hand picture, those squiggles are actually where the membrane of the cell binds in. And on those inner bits is where the factory is, where energy is made. And it's literally like a factory. So now I'll show you a cartoon of that. So that's the factory. And we have workstations. And essentially what happens is that we eat food, we digest the food and we metabolize the things. And then at the later stage, it goes into those energy producing factories. And the high energy molecules are fed in as the raw materials to energy production. At the left-hand end, called complex 1, they're passed along and we make our energy molecule ATP at the end. So that's how we make our energy. So I just want to show you a list of all the proteins that were dysfunctional or made in different amounts in the next slide. You don't have to memorize all of this. The green arrows say those that were made in higher amounts than normal compared with the controls. So we're using the controls. The red arrows are less. And the left hand is just the organelle, the mitochondria itself. And then we go through the various steps. And on the right-hand one at the top, the right-hand second to last one, complex 1, that's the first part of the factory, the first machine where the synthesis is occurring. And the bottom right is the last one where the energy molecule comes out. And you can see lots of proteins which are made in higher amounts. And again, what that tells us is the mitochondria, the body probably knows the mitochondria is not as working properly in terms of energy production. And so it tries to make more of the proteins for the complexes that are needed for the factory. So we're getting this interesting picture of the body trying to respond to your illness and do things that will alleviate it. But unfortunately, it's not quite as simple. So what we were excited, having got this, to say, well, can we then look at the functions of these mitochondrial complexes? Now, this is what I wanted to do back in 1990 when I put the grant into the Health Research Council that was turned down. But by now, we've got a machine called, it's a $300,000 machine called a seahorse analyzer, and we got one delivered to the University of Otago in 2018. And so that enabled us to look at live immune cells from patients and controls and look at how their mitochondria are doing. And I'll just sort of gently take you through a little bit of this. What we measure is oxygen consumption, and we can put drugs in as we're doing this, particular drugs which will inhibit things, and measure how each part of the factory is functioning. So for example, on the left, we start off by looking at just basal respiration. While you're sitting here, what are your mitochondria sort of doing in terms of just using oxygen to produce some energy? And then we can look at the actual amount of energy being produced. And the critical thing for ME is that what spare capacity do you have to produce energy if you have a stress event in your life? Healthy people have a reasonable spare capacity so that when you're stressed, you can actually produce more energy. That's a response to the stress. But if you don't have very much spare capacity, then you're not able to cope with stress so well. So we thought, well, maybe this is a critical thing for ME people. Now, when this analyzer was developed, and you could measure these things, a bioenergetic health index was developed for how well the mitochondria were doing. And so we firstly just looked at that. We did all of the calculations from our data. And what you can see on the left there, the MECF group is on the left, controls on the right. There's a scatter of data, but ME people generally come out sort of lower. And the theory is that if you've got healthy mitochondria, you've got lots of spare capacity, not too high a basal rate, and you can make ATP. If your mitochondria aren't working well, you've got a higher initial rate and low spare capacity. Now that didn't tell us too much. We wanted to look at the individual steps, and I showed this. So this is real sort of hard research data, and I just want you to look at the red ring thing. So in the middle column at the top is basal respiration. What you can see, the ME group, the line there, is higher than the controls, and that says the mitochondria are working harder. They're trying to work harder to produce their ATP. Just underneath that, there's the spare respiratory capacity, and there you see the ME group, the line is lower, and that means actually ME people don't have so much spare respiratory capacity to deal with stress. And then up on the top left is ATP production. So when we put the cells into our machine, they're not under the stress that you are dealing with real life. So the mitochondria, even though it has to work harder, can more or less produce the ATP. And you can see that line is only a little bit below the controls. Whereas if we really stress the mitochondria, and there's an Australian group that's doing this that I've been talking to, they show quite a big difference between the ability to produce ATP. So that was incredibly informative, and these were the two people that actually did this work in my lab. Okay, so I want to just come back then to those big questions I asked at the beginning. What's the control centers for the disease? Well, you might have thought from what I've just said, well, isn't it in the energy production? We think not. We think that's a really important consequence, but I firmly believe that it's centered in the brain, and I'll just tell you a little bit why we think that. And then so we ask, why don't people recover? We think because there is an ongoing stressor which doesn't turn off. And I'm going to show you, we think that's neuroinflammation. And so that would be the explanation. So that would explain not only not non-recovery, but it can also explain the relapse recovery. And if there's time, I'll just show you a little bit of the data with that. So this, we developed a neuroinflammatory paradigm, and I did this with a PhD student, Angus Mackay, who's an ME/CFS sufferer. Unfortunately, when he started doing experimental work, that was stressful enough for him to go into constant relapses. So what we did on a Friday afternoon, if he was well enough, he came and we sat down and we just talked. And we developed this theory together. And it's based on the hypothalamus, because some of you may know that the HPA axis, hypothalamus pituitary adrenal axis, doesn't function properly in ME people. We knew that. And we're actually within that hypothalamus, there's a sub-organ called the paraventricular nucleus, which just contains a cluster of neurons, which is your stress center. So if you're a normally healthy person and, you have a relationship breakup or something, then, you're stressed out about that. Your stress center would be dealing with it, and hopefully there'll be a flow-out effect, which I'll indicate one of that, which will protect all of your body from that stress. But we think what happens with ME is that there's cycles of fluctuating chronic neuroinflammation that can drive this stress center to be dysfunctional constantly. And that means you don't not allowed to go into a sort of a healing period and recover from the disease. So the normal homeostatic set point is not reset. And that, I think, is the key to curing this disease. If we can learn how to just set that point, then we'd get there. Now, a Stanford researcher, when we'd published our mitochondrial thing, contacted me because he was really excited because he thinks that damaged mitochondria actually act as a danger signal for the brain. So when the mitochondria have got more proteins than they're supposed to and they're a bit leaky, then now there's a growing body of thought that actually that acts as a signaling molecule. Just like in Alzheimer's disease, you may have heard of amyloid plaques. And so the amyloid is the natural thing in the brain that actually is an indicator of ongoing Alzheimer's disease. Okay. So I'm now at the last part of it, and I just want to tell you what we've done with the molecular control center in the cell, and that's the DNA. We wanted to go back to the DNA and see if we could get some explanations as why the metabolism was much lower, why these changes in the amounts of protein, where did this occur from? And you probably all know we've got a genetic code, and your genetic code doesn't change during your lifetime. This is the A, G, C's and T bases that you see on TV programs and whatever now. But while the reason we all look pretty well the same, we're all human, is because most of our genetic codes from person to person are identical. But we all have a little bit of variation from the person sitting next door to us, and that's why we're a unique human being and we look a bit different and whatever. There's another code that's only been discovered in the last 20 years, which is called the epigenetic code, and that's a dynamic code. And the easiest way I can explain this is there are little tags put on at all sites right across our 3 billion bases in our genome. And those tags are like light switches. They can be on-off light switches, or they can be dimmer switches. And so those are set so that all of our metabolism and all of the proteins in our body are controlled in just the right sort of amounts. And so what we wanted to do is to go back to ME patients and say, is this different in ME versus healthy controls? And I'm going to tell you the second study, we haven't published this yet, that we've just finished. And this is where we looked at the relapse recovery cycle, one of these big questions. So I asked 2 of our patients, university young women in my study groups, whether they'd be prepared to have blood taken every month for a year. And I said, there's just one other small thing. You have to guarantee to have a relapse during that time. And so what we were trying to do was to capture their normal kind of level of health, which of course is less than a healthy person, through a relapse and then a recovery to see what was happening in this molecular control center of the cell. So if you look in that we've got patient 1, patient 2 control. Hope you can read that. Patient 1, and I'm just showing, we decided to look at 5 of the samples on either side of the relapse. Patient 1 was quite well, and I told them to rate 1 to 10. So she rated at 7, so she was quite well to start with. In the next month, she rated at -3, ignored my instructions totally and didn't go 1, she went -3, but that told us it was a pretty serious relapse. The next one was -2, so she was still in relapse, the next sample, and then she went back to 7s. She didn't rate herself quite as well, it was satisfactory. The second patient was more fragile through the whole time, so her base level of illness was worse, so she ranked herself fragile 5 or 6, had a relapse down, she called 2. Probably, this is very subjective of course, but it was a relapse. And then the control, I had a obscenely healthy person in my lab who acted as a control. She ran marathons and things. She rated herself 10 through the whole experiment. And so what we did then is we looked at what we call the DNA methylomes, all of these the tabs through the genomes, and each of the samples, 5 samples from each person. So huge study, huge amount of data. And I'm just going to show you in this one result, unexpected result we got from it. From month to month, the healthy control showed about 1 site in 1,000 changing, a very low rate of change through the whole thing with her. But both ME patients were changing at about 2 sites every 100 sites. So 20-fold higher, if you like, variation in their epigenetic code. And that says there's instability in that code, which is going to change all of the body physiology that's occurring. So, you know, this has never been reported before and was sort of unexpected that it would be, you know, so dramatic. I'm not going to show the data. We were able to deduce all sorts of things, what were changing. But I want to now just show you the second study, one piece of information from it. This was a 10 patient versus 10 control study like the others. And what we found there was that there were 5 genes. Those are the red things with strange lettering in the right hand of the slide. Those genes are all involved in the release of neurotransmitters in the brain. And the neurotransmitters that the hypothalamus uses. So we think this is a second molecular signature for ME/CFS where there's changes to how neurotransmitters are being released and available to the hypothalamus. This will affect how that functions and affect the whole HPA axis. And of course, ME people have low levels of cortisol, which is regulating sort of the regulating hormone for stress at the bottom end. So that was a very exciting sort of finding. So I come back now to the jigsaw. Now I'm sure no one predicted that that was actually the jigsaw that was for that piece of puzzle. And that's really the problem we had when we started this. You know, we could see the bits of information and why I thought we couldn't just carry on with producing more bits of jigsaw, but rather we had to keep that big picture. And at least now we don't have such a clear picture as that one there, but we have a much clearer picture of what's going on in the physiology, I think, of the ME patient. So what are we doing right now? My last slide. In 2021, 2022, our plans. We've got a major study, in fact, the most major study being done globally on post-exertional malaise, which of course is the core symptom. So the first part was to do cardiac physiology with Dr. Lynn Hodges. And the interesting thing is there we've got 5 university women with ME as the patients. Each one of them shows a unique response in their cardiac physiology when they're put through an exercise regime. And what we did is we took blood samples before exercise, 24 hours afterwards. They did a second exercise and 24 hours after that. And we're doing a comprehensive molecular analysis if we can see what's happening to their molecular physiology as a result of that exercise. And so that's the project of Gemma Ellie on the top right. She was born into an ME family. Her father's had ME all of her life. So she's really excited to be doing some research to try and understand her father's illness. And then I've started a study on long COVID where I'm doing a comparative study between ME/CFS age match with long COVIDs to see whether the same molecular signatures that we've derived are present in the long COVID people. In other words, it doesn't look like ME/CFS, or does it have its own unique signatures? And I'm just involved in setting up collaborations with an immunologist, Dr. Anna Brooks at the University of Auckland, so we can add a new dimension to our whole study. So I'm doing all the molecular studies, and Anna is an expert in subfractionating and studying the subfractions of immune cells. And so she's totally committed to working with the long COVID group. She's part of the group. And she's got the same passion as I have for working with ME, so she's like a soulmate in that sense. So we're hoping, if we can get money and my health holds out for as long as Ros's has, that we can keep going and actually do our own studies on long COVID. So thanks very much for your attention, and that's the story.